Great. So I will introduce our speaker. This is very thankful to Kristina Ulicna. I'm just trying to pronounce that correctly. So Kristina is, she did her undergrad at King's College London after a BSc in biomedical science, during which she interned as well at Cancer Research UK and MIT. And she's now on the LIDo, the London Interdisciplinary Doctoral PhD Programme here at UCL. And she's working in single-cell tracking of heterogeneous cells in non-cancer and cancer cell populations. And she's going to be introducing us to some techniques around the area of deep learning that is used in cancer research. So I will hand over to Kristina.
Thank you very much, Patrick. Thanks a lot for the warm introduction, and thank you very much for having me. It's a pleasure to present this second masterclass, which is going to describe some of the introduction to deep learning. I know that I am speaking to quite a broad audience with a range of skills and range of expertise in deep learning. So one of the things that I wanted to do at the very beginning is to do some expectation management. I know that there are people, or there should be people joining us who do deep learning research for a living, and on the other hand, there are people who've never really trained any neural networks, but are really interested in the field. So what I wanted to do is to, in terms of the expectation management, please do not expect me to teach you how to train a neural network in like a 30-minute talk. That's almost impossible. Rather than covering the maths and all of the mathematical principles and the computer science behind the neural networks and deep learning in general, what I wanted to do here is to provide some real-life examples of how we in the lab use deep learning-based bioimage analysis and how it can help us in tackling very, very interesting questions in biology. So first of all, before I start, I want to introduce my data set, because this is what my PhD is aimed at, and this is the wet lab component of my PhD research. So what I do is that I do what's called live cell imaging. And if you're not a cell biologist, if you've never seen cells before, don't worry, I'll just briefly cover what it means. Live cell imaging intuitively says that we're imaging cells while they're alive, and we image them while they're growing. So what we do here is that we take an image or a snapshot of the cells at a certain frame rate. So in my case, it would be that I have an automated microscope and I take an image every 4 minutes. So I do this for a very long time, and what I can gain is then a time-lapse microscopy movie out of those images. And what it means is that we have a temporal resolution of 4 minutes, of the growth of the cells, and we capture them for a very long time. So that can range from 3 to 5 days, which usually comprises more than 1,200 frames. So in my time-lapse microscopy, the types of microscopy that I use are transmission microscopy and fluorescence microscopy, just as you can see on the slide. And I particularly am interested in 2 types of cell lines. One of them is of non-cancerous origin, and that would be the green one, or the MDCK cells. And the other one of cancerous origin is the HeLa cells. So here you can see on the left, on your left-hand side, you can see the transmission image, or a representation of a transmission image, and this effectively captures how the cells look under bright lights. And on the right-hand side, you can see a fluorescence microscopy image, which effectively tells you about nucleic acid organization in those cells. So these images we take in parallel, they're complementary to each other. And the way that the time-lapse microscopy, this type of time-lapse microscopy allows us to process data is with a very long temporal resolution. So we take these movies of a very big images for quite a long time. So here I want you to start thinking about how you can use deep learning in your particular area of research and how it can help you to tackle issues that you may face when you're trying to do automated data analysis and image analysis in your research. So here I drew 3 problems, or 3, well, let's say case studies that we try to answer using deep learning in the lab. So the first one is that sometimes we try to use deep learning when setting hard thresholds or cutoffs is very difficult. So what do I mean by that? So here on your left-hand side, I show you again my fluorescence input image. So this is the image that I get from my fluorescence microscopy, and I'm hoping that you can see my cursor so that I will point out to where you should actually be looking. So in my inputs, you can actually tell. So you can actually tell probably that the white blobs represent the cells, and the darker background actually represents just the plate. So there is no cells in that particular area. One of the things that we would want to do, or one of the problems that we're trying to address here, is that we're trying to detect and localize the individual cells per the frame. So what we want, as I try to illustrate here, is to get an output where all of the pixels that represent the backgrounds are scored as 0, so black, and all of the cells and all of the pixels that represent the individual cells are scored as white as the foreground. And also, what is very important is not just that we isolate these cells, but we also isolate them as individual objects. So we can then analyze them. We can analyze their size. We can study their position, and so on and so on. So this is hopefully you appreciate that this is quite a complicated problem, but one of the very intuitive ways of how you could approach this is to actually try to set a certain cutoff value, and you would say, okay, let's just say that I'm going to try to find a value of pixels below which all of the pixels will be scored as background, and above which all of the pixels will be scored as the foreground or the cells. So here you may want to do that, but hey, on this image. On this image slide, I tried to illustrate that it's actually much more difficult than it sounds in the first place. So first of all, the pixels on this fluorescence image vary, and they vary a lot because of the expression of the fluorescent tag which our cells have been transfected with. So you can see that there are some cells that are quite bright, for example, these in the middle, but there are some cells that are quite dim, for example, here in the edges, or even here in the center of the image slice. If you wanted to set a certain threshold for separating the foreground from the background, you may actually run into a lot of problems. And I tried to do this for you just to illustrate what it would lead to. For example, if you set a fairly low value of, say, 70, 78, you would actually segment this one huge blob, this one huge, gray cell, and it would be scored as one entity, as one object. So that would be one cell. Now, you can probably tell that is not correct, and the segmentation in this case actually fails really badly. So you can go and try to increase the value of the pixels. For example, you select a 137, as in here, in this cell. And, well, you can see that you're getting towards a better result. You can see that some of the cells actually get segmented, some of the bright cells get segmented, but most of the dim cells are actually forgotten by the thresholding, because they're just, their pixel values are below the threshold. What you can also get when you do this sort of thresholding is that you get a lot of single pixels that are in the background, which now would be classified as cells. And again, that's incorrect. The other extreme is that you go to a super high value. You go to almost white pixels. So you would, for example, select a number of 216. And what you see here, one of the things that you see here is that you're only really getting the brightest cells, and most of the cells and most of the background is not classified or segmented correctly. So one of the things that we tried to do in the lab is that we used a deep learning approach. And if you're familiar with the architectures that people use who work with deep learning, in particular, to target this particular example, we use the unit architecture, which can do this for us. So it takes a fluorescence image input that is relatively noisy, and it translates it into a binary mask, or a mask that segments the individual cells into individual objects which are placed on the background. Now, The other case study I want to demonstrate here is that sometimes we try to use deep learning when manual annotation would literally take a lifetime. And what do I mean by that? Okay, so for those cells, now we have a different problem. We now segmented those cells, so we know where they are in terms of their localization coordinates. But we also want to ask the deep learning model or any other model to assign every cell in this slice a classification label. And the classification label would tell me about the cell state or how far it has progressed towards mitosis. If you're not a cell biologist, if you've never seen a cell divide before, don't worry. This is what I'm showing you here in this panel. So what you can see here is that a cell that is an interface, which effectively means is not dividing, if that cell approaches division or makes a decision to divide, it actually undergoes a sequence of morphological changes. So its nucleus and its nucleic acid actually gets reorganized and gets is now in different shapes and in different pixel intensities. So here, hopefully, you can appreciate that setting the hard boundaries between these labels, between these states, is actually not difficult. Hard boundaries are actually super easy, because I can tell exactly from these image crops whether the cell is an interface or whether it undergoes division. So it goes through prophase, metaphase, anaphase, and then it restores itself back to interface. The problem here is, as I illustrated already, is that if I wanted to annotate every single cell in all of these input images by myself, by hand, and if I wanted to pick or score those cells based on their face, it would literally take me forever to do that. And so instead, we're not really relying on the manual annotation here, but what we've done in the lab is that we trained a deep learning model which uses convolutional neural network architecture, which can actually do this for us. And the way it works is that it actually crops a tiny image around the cell, and it uses its corresponding bright field image together with the fluorescence to actually decide which label the cell should be assigned with. And what this actually means is that it displays the probability, the highest probability of that particular label per cell. So what we do here is that really we're saving a lot of time. And not just that we're saving the time of the student, but we also can get these outputs really fast. So a single processing step of a combined segmentation together with a cell state classification for a movie that is comprised of 1,200 frames or maybe even more takes less than 5 minutes. So this is a super fast approach to actually annotating images. The third case study, and I must say this is the one that is the most intriguing to me, and I find this really fascinating, is that deep learning sometimes helps us to solve problems that humans cannot solve. And to illustrate that, I try to pick an example which represents one of the future objectives of what I want to do for the rest of my PhD, and I'll try to lead you through it. So here we're trying to answer a so-called regression task, and I'll explain what that means. So what you can see here on the left-hand side is that you see a transmission image, a bright field image of a transmission microscopy, which captures growing cells. So one of the things that you can hopefully tell is that every single cell on this image looks different. So this one is, bigger and round, and this one is super flat, and you can't even properly see it. And those they seem to have a higher contrast, and this one is probably undergoing division because it looks kind of like it's dividing. But you can hopefully appreciate the diversity of the visual appearances and of the morphology of those cells. Now, one of the things that I'm trying to ask here is, can you tell, based on just the visual appearance of those cells, can you tell how long each cell is going to take to divide? Now, this is, in fact, a regression problem. So what we mean by regression is that we're trying to train a deep learning model or an algorithm that can regress or predict the cell cycle time. And the way we do that is that we again supply a tiny image crop that captures the cell. The cell is right in the center, and we also provide the answers. Or the labels to those images. And in this way, we can actually train a deep learning model, or well, I'm attempting to train a deep learning model, which could tell me based on the features in these image crops. Now, this is something that may be very difficult to actually understand and to grasp the idea behind, because you can start wondering, okay, how is it possible that for humans, even if you showed me millions and millions of these low contrast examples of this image example, I would not be able to tell. I wouldn't be able to learn when to tell the cells when to properly predict the cell cycle time. But computers are actually really well suited, and especially deep learning algorithms are very well suited for tasks like this. So how is it possible that it can tell, it can target, and it can address problems that humans can't even solve? Let's try to answer that together. So again, I'm coming back to my fluorescence image example. And here, again, we're back in the task of segmentation. So what we want the computer to do is to clearly just mark where cells are and where cells aren't. So separate the foreground from the background. Now, if this is my data set, so the example of the data that I supplied to the computer, even if we zoomed into a really dim cell, you don't necessarily need to be a rocket scientist to be able to circle around these cells. It's actually pretty obvious. And I'm pretty sure, a 5-year-old child could do this fairly easily. That is because we humans perceive information with its context. We perceive so-called contextual information. So for us, the combinations of the pixels in this zoomed-in image actually mean that here the brightest and the brightest circle represents a cell, and these images that these pixels that don't tend to be that bright are actually backgrounds. However, to appreciate what computers have to do, we really need to understand how computers perceive images. For a computer, I tried to illustrate it here on the right-hand side example. For a computer, it's very, very difficult to actually perceive context. And that is because, in fact, the only thing that the computer is able to see is that it sees a matrix, or, if you will, a table of pixel values. So these are just numbers for the computer. That's the only thing that it sees. And actually, these two images, hopefully it's quite clear that they're corresponding to each other. So now, I actually dare you to try this yourself. If you wanted to understand how difficult it is for a computer to actually segment images, just try to convert a single image, a single pixelated image into this pixel matrix, and now try to circle around where the cells are. And hopefully, by doing this exercise, you'll understand how difficult, and you'll appreciate how difficult it is for a computer to make conclusions. So if you were a computer, let's imagine that we're all computers at the moment. So if you were a computer, you actually have a few strategies of how you can target this problem. So one of the things you can do, it's not very favorable, but you can certainly do it, is that you can memorize all of these answers. So what I mean by that is that you can store somewhere in your computer memory, you can store all of these values of the matrix, or the table of these values. You can store it in memory and memorize that every single time this particular example comes to place, I'm going to say, okay, a cell is here. Now, you hopefully see that this is not a really good approach, because what computers, because, in terms of memory, that you probably couldn't, handle that much information. But also, if you had the capacity to store that information, it still doesn't give you much of an advantage. And the reason is that even if you change a single pixel value in this image, now your computer wouldn't be able to make right predictions, because it would never see that example before. So it would see something similar, but it wouldn't be able to say that, okay, this is similar to what I've seen, because the values would not be identical, and the values would be completely different. So sometimes what we say when computers try to memorize the answers that it's seen in the training data, we say that the model is overfitting. And that means that it performs really well on the data that it has seen before, but as soon as I change it a little bit or I provided data that it hasn't seen before, it fails to make correct predictions. So what we really want to make computers be able to do is to learn the patterns. And what we mean by learning, this is the learning, you know, everybody mentions deep learning. The learning component is really important. But what we mean when we talk about learning is that we want the computer to understand or appreciate the patterns and the correlations of the pixels that represent the patterns. So for example, here in this particular segmentation image, we really want the computer to be able to understand and internalize the behavior of those pixels together in that image. When we achieve that task, we actually can extend these models to tell us something about data it hasn't seen before. And if it's able to still make correct predictions on those data that it hasn't encountered yet, we say that it's able to generalize well, or that it's capable of generalization. And that generalization effectively means extending the knowledge which you inputted to the model and getting something out of it on data it was not trained on. Now, this is super cool, I think, but what we need to keep in mind is that it's not enough just to show the computer a single image with a single cell circled around. What we actually need to do when we train models and when we try to make computers learn patterns, we need to actually gain large data sets, and we need to provide that to ensure proper training when it comes to our deep learning models. So yeah, this is one of the limitations of deep learning, and that is that if you only have a few observations of a certain behavior, you're probably not going to be able to train your model effectively, and you'll rely on overfitting. So your model will only be able to memorize your data, but that's not really what you want. What you want is for the model to be able to generalize well on other examples for the future. So you may be wondering, okay, deep learning is something that is really popular nowadays, and in the last, 10 to 5 years maybe, you've probably learned a lot about it. But deep learning is not the only approach that we use to actually analyze data, and definitely not in a sort of like bioimage analysis field. It's definitely not the only approach that we can use, and I think it becomes very clear what the real power of deep learning is when we start comparing it to the more traditional methods, the more kind of old-fashioned computer vision methods. And so here on this slide, I actually try to contrast just that. So when we think about the more traditional computer vision methods, what we have to do is that we provide the algorithm with the problem. So for example, here it would be the image crop of my cell. And I'm trying to answer the question of how long would it take to this particular cell to divide. But what I want to do, what I have to do when trying to address this problem is that I would have to input some sort of features or some sort of characteristics that I, as a scientist, believe are important when determining the cell cycle duration. So for example, one of the things that I would have to do is to, say, extract the shape of the cell. So you can see here that the cell is fairly round, and I need to use some sort of mathematical algorithm that are usually very complicated and actually quite laborious to design to perform some sort of edge detection, or I have to do some image histogramming, or I have to do some sort of witchcraft with the computer science to actually be able to extract that information. And once I build those features with my previous hypotheses of what I believe is important when determining the cell cycle control and the duration, then I can ask the computer to provide the answers. So here, what is very important that the knowledge of the rules is required, and the knowledge of the features and what is important in that image, that is something that I require to use for computer vision. However, deep learning doesn't necessarily rely on this, and actually it can do something even cooler than that. So in deep learning, instead of providing the computer with the problem and the features, we provide a computer with the problem. So we, just as before, we input the image data, and then we also input the correct answer. So the correct answer in this case, for example, would be 8 hours and 56 minutes. So and that is the time that it would take the cell. That is the time that it would take the cell to divide. And really, what the power of deep learning is that it can couple these problems with these answers in a very efficient way, and it can come up with the features that are important to connect these data to these answers automatically. So it can automatically figure out the patterns, and it can dissect what exactly is it that it's important to make that prediction. It can dissect and it can tell what exactly tells me that this cell is going to divide in this range of times. Now, this is really great, because what this allows us to do is to approach problems in a hypothesis-free way. So what it means is that the complete ignorance of the rules is allowed here. And what I mean by that is that we don't need to know, I don't need to have any scientific evidence or any hypothesis that that tells me, okay, I think it's the shape, or I think it's the roundness, or I think it's the intensity. I can approach this problem in a hypothesis-free way. And the deep learning model, if trained well, is going to automatically figure the rules out. And this is, even though I'm presenting it as a great advantage, it can also function as a great disadvantage. And what do I mean by that is that sometimes deep learning, I try to illustrate it here. Sometimes deep learning is referred to as the black box, or some sort of magic, or some sort of witchcraft, or it's actually really difficult for us scientists to understand what exactly the deep learning model is looking at. Even though it can do it, we don't understand what it's looking at. And this is where the issue of interpretability comes to place. And this is something that I think represents one of the future challenges, and even current challenges, of the deep learning research. So to make you understand what I mean by interpretability of the results of the deep learning pipelines, I really wanted to illustrate this difference between the supervised learning and unsupervised learning. And what I mean by that is that we really need to understand the difference between the two approaches. So far, in all of the examples that I've shown you already, I was approaching my problems in a supervised manner. So what that means is that I provided my initial data, or my problem that I tried to solve. So again, in this case, it would be the images or the image crops of the cells, of the growing cells, and I also provided with the correct label. So the label, what it means is effectively, this is the correct answer that I have, that I know is true. And I want my deep learning model to connect these two. I want to find the connection between the data and between the labels. And if I provide enough, the sufficient amount of the data and sufficient amount of the corresponding labels, I can assume that my deep learning model will actually internalize those behaviors and it will learn. The problem, however, as I already illustrated, is that we as humans don't really understand what it's looking at. Even though it works, which is amazing, we can't really dissect it on, we can't really dissect it in details, and we don't know how exactly it's making these decisions. And to make you understand that even more, let's consider this example. I provide my data, right? I provide my tiny image crops, and then I provide the number, say 15. But I don't really tell the model what that 15 is. It can be, the cell cycle duration. It can be, how heavy the cell is, or it can be, the number of, likes on Instagram. It doesn't really know what I'm looking at. It only connects the numbers to the initial data. But if we want to understand it, for example, in medical applications, if we really want to understand it, or even here in biology, if we want to understand what exactly it is that drives the cell to divide at a certain time range, we really need to be able to interpret these machine learning and deep learning models. So I think a nice way to start thinking about is to actually understand what unsupervised learning is used for. So In contrast to supervised learning, when we try to train a model in an unsupervised way, the only thing we provide is the data, and actually we provide a lot of it. But in this case, and now we do not provide any labels. I do not tell my model the right answers to my problems. So the only thing the model is left with is a lot of data. And it may be confusing to actually grasp the idea behind this, but the only thing that the model is left with at this task is that it starts to look at the patterns and it starts to look at characteristics that are similar in those data sets. So one of the methods that people use very often in unsupervised learning approaches is so-called clustering. So this is what I try to illustrate here on these examples. So in the first cluster, the cluster number 1, you can see that the cells are grouped together based on certain characteristics. And those characteristics should be different to cluster number 2. And so if you spend time, looking at those cells, something that I try to make it very obvious here is that the cells in the cluster number 1 are actually more round, and you would say they are more kind of regular than compared to the cells in the cluster number 2. And just a visual inspection like this, a simple visual inspection, can actually tell you a lot about the behaviors of those clusters. And now you can go back to your raw data and you can start analyzing, okay, well, this cluster, if a cell is round, it probably means, the round cells will have something in common compared to the, spindle-shaped cells or, to the irregular cells. And here, as I said, I tried to make it very clear and very obvious how this clustering happens. But when you actually address complex data sets, sometimes your deep learning models can give you answers to less obvious solutions. But unsupervised learning can actually help you a lot with the interpretability of the models. And I just again want to point out that this is one of the biggest challenges in deep learning research, because just training models that work well is not enough. We also want to be able to understand why it's making predictions, why it's making them, how confident it is, what it makes them, and what drives the model to make those particular predictions. So before I finish off, I just want to do a little promo of an upcoming paper of things that we've actually done in the lab to try to address some of these issues. So, I introduced some of the segmentation and then some of the classification of the cell states. Oh my God, it's so complicated. Why would anyone do that? And the answer to that is hopefully presented here in the snapshot of the figure from the upcoming paper. So what I tried to illustrate here is that we actually, in the lab, we spent some time to develop, a lot of time actually, a lot of time to develop these cell annotation techniques, or that comprise of the nucleus segmentation and the cell state classification, just as I showed you. And now, because we have these long movies that I introduced at the very beginning, we have these long movies, and we can now start following the behaviors of particular cells. And when you follow something, when you follow something over time, you usually call it tracking. So this is exactly what we work on in the lab. So we've developed these single cell tracking algorithms that can follow and connect the contextual information from individual frames over time, just as I illustrated here, on this figure. And what this allows us to do is to actually, in a very simple and automated way, we can now extract the single cell lineages and visualize the lineage trees. So for the 4 cells that you have here in this image, their lineage trees, or parts of their lineage trees, would look something like this. So we can now account for the cell movement, for the migration abilities, and also we can now record the division history of exactly when individual cells have divided. And what this tells us, allows us to do is to actually, decipher and dissect a lot of molecular and neighborhood influences that actually influence and impact the cell division times in our populations. So this is something that I've done during my PhD, and hopefully you stay tuned for the upcoming paper. You can hopefully in a few weeks you can read more about this if you're interested on bioRxiv and farther on. And yeah, I'd like to leave you to it. First of all, before I finish, I want to wholeheartedly thank my supervisors, Guillaume Charras at the London Centre for Nanotechnology, and Alan Lowe, who's based at the Institute of Structural and Molecular Biology, both at UCL, and their group members for being wonderful colleagues and even better friends. And, if you're interested in more, please follow me on social media. Hopefully I made it quite clear on how to, reach me online during the presentation, because I'm gonna share my slides so you can download them. And I'm gonna also share some links for interactive deep learning courses that really helped me to actually learn about deep learning when I started doing my PhD. And of course, to stay tuned for some further research and for some further updates of what we can do in terms of bioimage analysis when using deep learning. So thank you very much, and I'd like to take any questions that you might have.
Thank you, Kristina. That was really interesting, fascinating. It sounds like it's got a huge amount of potential for cancer research, these sort of techniques.